Okay, this is the first of the virtual lectures that we'll be discussing dihybrid crosses. Um, Make sure you have a pen or pencil and some paper because as we go through this, I'll be guiding you through a practice problem to make sure you know how to do this. So have that handy. You can always press pause and come back to the video. You can always rewind or watch this again if you need to. Last week we left off by doing some monohybrid crosses, which were crosses involving only one trait. And in this lecture, we'll look at dihybrid crosses, which are crosses involving two traits. Once again, di means two, and hybrid is a combination. So two traits are being combined in these crosses. There are 4 steps that you would have to go through in order to complete a dihybrid cross, and it's similar to the method used for a monohybrid cross. The first thing that you must do is determine the alleles that are produced for each gamete of each parent. Then you would construct your Punnett square with the appropriate number of boxes. With dihybrid crosses, your maximum number of boxes would be 16, so a 4x4 square. It doesn't always require a 4x4 square, but if that makes you more comfortable, go ahead and do that. Then you would have to combine the gametes within the Punnett square, and then finally interpret what those gametes stand for in order to calculate your genotypic or phenotypic ratios. In the last step, it depends on what the question is asking, whether you would compute genotypic or phenotypic, or both. Again, your focus is to interpret what's inside the chart, what the dihybrid cross is telling you, and answer the appropriate question. In order to determine the alleles, it's a little bit like the FOIL method from algebra. So if we're going to use an example from Mendel, let's consider P color and P shape. We're using the letter Y to represent yellow, which is the dominant trait, and the lowercase version to represent green, the recessive trait. The letter R, the capital, represents round, which is the dominant shape, and lowercase r, which is the recessive shape, represents wrinkle. In order to determine the gametes that are possible for an individual, you have to know the individual's genotype. There are going to be 2 sets of letters since this is a dihybrid cross. The first set of letters represent the first trait, and the second set of letters represent the second trait. So for the first example, if there are 2 capital Y's and 2 capital R's, the only type of gamete that can be produced is 1 capital R and 1 capital Y. Remember that a gamete has half of the genetic information of the parent. Gametes are haploid compared to all the other body cells or somatic cells being diploid. Look at the third example, where there's a heterozygote for both traits. Capital Y, lowercase y, capital R, lowercase r. In the FOIL method, FOIL stands for first, outer, inner, last. So if we gave each of the letters a number, the capital Y would be a 1, the lowercase y would be a 2, the capital R would be a 3, the lowercase r would be a 4. FOIL shows us that we combine 1 and 3, 1 and 4, 2 and 3, and 2 and 4. The heterozygous individual for both traits would give us the maximum number of gametes for a dihybrid cross, which is 4. And they are capital Y, capital R, capital Y, lowercase r, lowercase y, capital R, and lowercase y, lowercase r. There's actually a typo in the PowerPoint, and I just realized that. When you're constructing your Punnett square, this is the second step. Remember that you would need to have 1 space for every individual unique gamete. If we consider the cross where the parents are both heterozygous for both traits, that's the only time that you would need to use a square that is 4x4 with 16 boxes. It doesn't matter which parent goes in which space, but what's important is that you use the appropriate number of spaces. Remember, meiosis produces 4 unique daughter cells, but sometimes in the dihybrid cross, those 4 cells might have the same information for just those 2 specific traits that you're tracing. The third step of doing a dihybrid cross would be combining the gametes. So pictured on the right is the cross where both parents are heterozygous for both traits. So you see the gametes across the top represent parent 1, and the gametes across the side represent parent 2. You would have to fill out the squares with the alleles from both parents, similar to our monohybrid Punnett square, except keep the similar letters together. For example, the square in the upper left-hand corner, you would drop down the capital Y and capital R from the top and push over the capital Y and capital R from the left, so that you would fill that in with 2 capital Y's and 2 capital R's. For box number 2, which is at its side, the same thing. You're going to drop down the capital Y and lowercase r and push over the capital Y and capital R so that you have 2 capital Y's and then a capital R and lowercase r. In this case, these individuals still show the same phenotype even though they're slightly different genotypes. So let me go ahead and just fill in the rest of this chart here. And realize that each box you have to look at to determine what's the phenotype of that box. Since we're dealing with 2 different traits, and at this point each of those traits have 2 different features, there's a possibility of 4 different outcomes that could appear in this chart. Since Y stands for the color, either yellow or green, and R stands for the shape, either round or wrinkled, our 4 choices of phenotypes are yellow and round, green and round, yellow and wrinkled, and green and wrinkled. So that's what you would have to do at our very last step, which is calculating the ratios. Yellow and round would be anything that is both dominant. If you look at the chart on the right, you would be able to notice that there are 9 different yellow round genotypes in the dihybrid Punnett square. The next 2 phenotypes, green and round and yellow and wrinkled, both show 3 boxes with genotypes that fit that requirement. And finally, the box all the way at the bottom right represents the 1 individual, or 1 out of 16 chance of ending up with a green and wrinkled offspring from the initial parent cross. In this case, where both parents are dihybrid for both traits, the phenotypic result would be 9:3 to 3:1. Again, 9 represents 9 out of 16, 3 out of 16, 3 out of 16, and 1 out of 16. In this case of a dihybrid cross, it's a little unlikely that you'd be asked the genotypic ratio because that's a bit more complex. What's most likely for me to ask you is the genotypic ratio according to the parents. And if you remember, the parents were both heterozygous for both traits, so capital Y, lowercase y, capital R, lowercase r, and there happened to be 4 individuals in this square that fit that. But again, you would have to calculate the ratios based on what the question is asking. So now it's time for you to practice. In pea plants, purple flowers, capital F, are dominant to white flowers, lowercase f, and tall stems, capital T, are dominant to short stems, lowercase t. If a tall, heterozygous, purple, homozygous, flowered pea plant is crossed with a short, white flowered pea plant, what are the possibilities of having offspring of tall and purple, tall and white, short and purple, short and white? So now follow those 4 steps in order to do this practice. So I recommend that you pause the video now, create your Punnett square, create the gametes, and then check to see if your answers are correct. I'll start you off by saying that the first parent If it's tall heterozygous, purple homozygous would be capital F, and tall heterozygous would be capital T, lowercase t. Sorry, I jumped the gun on that one. So again, the first parent is tall heterozygous, capital T, lowercase t, and purple homozygous, capital F, while the second parent is short, lowercase t, and white, which is lowercase f. You can pause now and try your Punnett square. If you haven't paused, you'll hear the answers right now. 8 out of 16 of the individuals, or 50%, would be tall and purple, and 8 out of 16, or 50%, would be short and purple. From this cross, there is no way to end up with individuals that are tall and white or short and white. This is a case where you wouldn't have to use the Punnett square with 16 boxes, because some of them would be repeats. There are some review questions here in order for you to review dihybrid crosses. If you didn't get those answers to the last set, I recommend that you redo it in order to understand. Part of the take-home quiz is going to have you answering your own dihybrid crosses. This is the end of the first of the online lectures.